So I'd like to welcome everybody to the 8th annual Oliver Smithies Symposium. This was made possible by the generous gift from Oliver, and he stipulated that one of the goals that he wanted to achieve was that young scientists get a sense of eminent scientists and the discovery process, the excitement, the disappointment, etc., that goes along during their careers, have gone on during their careers. So Oliver was a member of our department for 25 years. He really is known for 3 bodies of work. First, he developed starch gel, or refined starch gel technology, and he did that because he was interested in looking at proteins in blood. And that in and of itself is obviously something of huge impact in the whole field. He then collaborated with Fred Blattner, and they really pioneered the sort of genomics revolution because they developed the technology to allow cloning of large pieces of DNA, that developed the technology to sequence and to analyze that information. And finally, of course, he's known for developing the technology to genetically modify mice, for which he was awarded the Nobel Prize. Whoops, sorry. Oliver had a few traits. I actually was a graduate student here at the time. I was lucky enough to listen to Oliver talk. First of all, he was all about good science. And he talks about his PhD paper that resulted from his PhD work with great pride, even though it's a paper that's never been cited by anybody else in the world, including himself. Oliver had a real passion for science, and he taught a course that I took, and it was a lecture course. He would come running into the room 3 minutes late. He would have a stack of reprints, because this was pre-internet, pre-PubMed. And he would then proceed to talk about those 8 or 10 papers for the duration of the lecture. And it was obvious how passionate Oliver was about the papers he was talking about. And these were, generally speaking, new work that was just been published, and the context in which it was important. Oliver was a consummate teacher. Not only the passion that he brought to the courses that he taught, but he actually was thinking out loud. In retrospect, I realized that all of the major parts that allowed genetically modified mice to be made, he talked about in his lectures. He was basically thinking through the literature. And one of I think a great metaphor for Oliver is what I call the runway moment. And as some of you know, he was a pilot. He liked to fly planes. And he described this sort of moment when you break through the clouds and everything has been calculated correctly and the runway is in front of you. And he likens the discovery process to that moment. And in fact, he writes about how when the southern gel the autorad for the southern gel that showed that the targeted piece of DNA had gone into the right place, that gene targeting had occurred. He likens it very much to this moment in time. So finally, I want to thank the sponsors who make this symposium possible, and I want to introduce Dave Paglarini, who really organized the speakers this year, and he's going to introduce the speakers. So, Dave.
Great. Is he going to wear this, or is he going to have his own? Okay. Great. Okay, so it's really exciting for me to be introducing these three speakers today who have made profound contributions to my favorite field, that of mitochondrial biology. It's especially exciting for me to introduce the first speaker, Professor Vamsi Mutha, because he was also my postdoctoral advisor. So Vamsi grew up in Texas and lived there until he went to Stanford, and where he majored both in mathematics and in computational sciences. He then went to Harvard Medical School through the Harvard-MIT Health Sciences and Technology program, and where he started doing research in mitochondrial bioenergetics with Bob Balaban at the NIH. After an internship and residency at Brigham and Women's Hospital in Boston, Vamsi then did a postdoctoral fellowship with Eric Lander, then at the Whitehead, who is now the director of the Broad Institute. So Vamsi joined the faculty at Harvard in 2004. He's also a member of the Broad Institute. So Vamsi's group, true to his training, really combines clinicians, computer scientists, and various types of biologists really in an interesting, synergistic, powerful way. I think he and his group have a real special ability to harness the power of large-scale data sets and to use those to really come up with honed hypotheses about mitochondrial functions. And this is evidenced by some of the major discoveries he's already made, including identifying over a dozen Mendelian mitochondrial disease genes, discovering that mitochondrial dysfunction is associated with type 2 diabetes, had a lot to do with figuring out the componentry of mitochondria, what the proteome is, something I had the honor of taking part of as a postdoctoral fellow. And also recently, identifying the mitochondrial calcium uniporter. This was something that was known in the field for more than 50 years, but the molecular identity of it was not known until some of his very recent seminal work in the area. So Vamsi has won quite a number of awards, including the MacArthur Fellowship Award, the Genius Grant. He had the Kalin Medal from the Biochemistry Society. He's recently inducted into the National Academy of Sciences and was named an investigator in the Howard Hughes Medical Institute. He is also a state tennis champion in Texas, and he tells me his top serve was clocked at 109 miles an hour. So I guess that's pretty good. In addition to that, Vamsi is just someone who is very excited and optimistic about science, and was and has been really a fantastic and supportive, inspiring mentor. So I'm really excited to welcome him here to Madison. Vamsi. Had to work it in.